Hello everyone, my name is Shiniti Nagai. Currently I'm a PhD student at Institute for Computational Genomics from Aachen Hospital University, and today it's my pleasure to present our work dissecting differential cell-cell communication with CrossTalkeR. One specific cellular crosstalk occurs through ligand receptors, where cells secrete ligands inducing other cells that have its cognate receptor. Here we have an illustration showing how the ligand receptor analysis is done using single-cell RNA-seq data. As an input, we have a ligand receptor database, a single-cell RNA-seq matrix, and the cell type information from the single-cell RNA-seq analysis. In this way, the methods looks at the gene expression. As example, we have the green ligand expressed on the purple cell, and its cognate receptor expressed on the red cell, indicating that it's a possible interaction to be recovered. This process is done using all possible ligand receptors from the database and all cell types from the single-cell RNA-seq matrix. Even though we have several methods that does the ligand receptor analysis, we have some limitations. So usually ligand receptor analysis returns large number of interactions. It's not possible to compare changes across different biological conditions, and network properties are mostly ignored. Here we have two methods. One is the CellFoDB, is a most adopted method to perform ligand receptor analysis. It considers complex of receptors and it does the permutation test over using ligand receptor pairs in different cell types to identify the most reliable interactions. On the other hand, it does not allow us to make a comparison between phenotypes and do not explore networks. CellChat is another tool. CellChat explores topology and topological measures in networks using single cell and cell-cell data. All the analysis in CellChat is done using signaling pathways, usually retrieved from Craig, and also CellChat considers the complex of receptors. Just to point out, there are some receptors that have multiple subunits, and these multiple subunits is assigned to different genes. That's why it's important to those methods to consider that. Even though CellChat allows us to obtain some comparative measures, the network topology is not fully explored to do that. To overcome with this, we introduce and we developed CrossTalkeR. CrossTalkeR is a framework to prioritize and characterize changes in crosstalk using ligand receptor analysis data. It's an R package, and to do that, CrossTalkeR builds two network resolutions. One prioritizes cells and another focused on the gene cells. Here we have also the use of topological measures to do the prioritization, and in the end, CrossTalkeR produces an output in a format of a shareable report, and all the data is stored in a R object to allow a reproducible research. In the initial step, CrossTalkeR takes the ligand receptor interactions, usually from CellFoDB, and builds a gene cell network. In the gene cell network, each node represents a gene cell, and the gene here can be associated either a ligand or a receptor. And if the receptor is cognate, then we have an edge. Note that the edges always occurs from ligands and receptors, although we can have some back edges in the case that the same gene or same protein is associated to either a ligand and receptor. Another resolution that we provide in the CrossTalkeR is the cell-cell network. To do that, the gene cell network is contracted to keep only the cell-cell information. In this way, all the interactions given a cell pair is aggregated by sum, as shown by the interaction W1 and W3. That is the sum of the interactions from the gene cell network. Once we have these networks, we use topological measures to do different prioritizations. So we state and rename some some measures to allow an easier interpretation and also to make all the rows or all the prioritization clear. Influencer is implemented using out degree, listener is implemented using in degree, and here we have mediator, which is implemented by between the centrality. By using those measures, we can identify the players that are sending more signal, receiving more signal, or even the players that are doing the bridge between different players. We also assign a node importance using PageRank. In this case, the PageRank is a measure which is obtained through random walks in the network, and using that, it returns a probability vector for each node. The differential analysis here is done using those measures. So we calculate those measures for each biological condition. So as example, we calculate these measures for control and the phenotype one. And in the comparison step, we use control as a reference, and then we can have the information how the phenotype 1 is changing towards the control. Because of the PageRank is a vector of probabilities, here we are using the log odds, which is written here as a subtraction. To illustrate how CrossTalkeR can highlight the important players in cellular communication, and also to validate our methodology, we applied it in the bone marrow myelofibrosis single-cell RNA-seq. Here we have a schematic made by Kramer and Schneider back in 2018, which shows the main changes in myelofibrosis disease. As you can see, we have a down regulation or down expression of CXL12, which is a hematopoiesis marker. We have an increase of inflammation indicated by TGF-beta 1, also CXL4. Moreover, we have presence of a myofibroblast and dysplastic megakaryocytes. By looking this, we have some open questions. So we would like to understand which cells are producing those myofibroblasts or inducing, and how the fibrosis is starting in the bone marrow niche. In attempt to tackle these questions, in a collaborative work with Schneider's lab, we did a single-cell RNA-seq. Here is a single-cell RNA-seq for patients. We could retrieve 5 different cell clusters. We can see that the differences in the markers shows that the data really represents each phenotype really well. And in the first step, as I said before, CrossTalkeR builds those networks. So here we have the cell-cell network, and the nodes represents each cell type. The node size represents the page rank of the node. The edges represent the aggregated sum of all ligand receptor interactions. The edge size represents the number of interactions between those cell pairs, and the colors is the aggregated value of the expression of each ligand receptor interaction. In this way, we can see some difference, mainly the loss of fibroblast and MSC interactions in the disease, also a slightly upregulation of megakaryocytes of interaction, but by looking at it, it's really difficult to discern the real difference. In this case, we provide to the user a differential plot where we can highlight the up and down regulation towards the disease, and the nodes represents the log odds of this node importance given by the log page rank. In this case, we do see some results that match with what is expected in the disease, such as the increase of megakaryocyte signaling towards different cells, mainly towards MSCs and neuronal cells and fibroblasts. Another representation is the cell gene network. Although we have this graph, it's really difficult to interpret. That's why we use all the measures calculated by comparative step to build a PCA embedded space, and we project all the nodes in this space. In this way we can identify the nodes that are related to each row, as we stated before, and also the nodes that are related to this biological condition of interest. In this case, we see the increase of collagen 1 in MCs, indicating the production of matrix, or the extracellular matrix to be more specific. We have increase of TGF-beta 1 in megakaryocytes, a loss of CXL12 on MSCs. Those markers indicate matches with the schematic that I showed before, and also it was really interesting to see that CrossTalkeR were able to use those measures to highlight those players. We also allow the user to go deep on each of those interactions. In this case, if we look at the TGF-beta 1, here we have most of the TGF-beta 1 interactions, and we do see that the interactions that comes from megakaryocytes is always upregulated, but we have some downregulation looking to the MSCs, neural cells, and fibroblasts. And the idea here is just to provide to the user a small list about the nodes, the selected nodes. To wrap up all these ideas, we have that CrossTalkeR were able to highlight the main myelofibrosis communication players. CrossTalk is a framework to prioritize and characterize cell-cell communication using two network resolutions. It can be done, or it can be used within a single phenotype or cross phenotypes. CrossTalk allows a shareable and reproducible research by using standard output formats and objects. We recommend to look at the sample of the report available in your GitHub, and also if you have the opportunity, take a look on our vignettes that are available on our GitHub page too. As an ongoing work, we are attempting to fulfill our network with the intracellular data too, using single cell ATAC-seq to predict the transcription factors and connect it to our ligand receptor networks. We are also attempting to obtain statistical significance for those differential interactions, and we are also investigating the possibility to use network diffusion to highlight patterns related to the specific conditions. CrossTalkeR is a team effort. I would like to acknowledge and thank you Niels, Rebecca from Cell Biology Group, also Michel Schaub for Computer Science, Ivan Costa for Institute for Computational Genomics, and all the people that provide some inputs to make it possible. CrossTalk is available on GitHub. We have also a documentation. I'm tracking the issues frequently. I would like to thank you, all the organizers and the staffs that makes the conference possible, and thank you. I'm here for questions.